Great. So hello, everyone. I do not speak a lot of Spanish, so my presentation is going to be in English. I would like to excuse myself for that. But for the last, I would say, 36 hours, I have been listening to some excellent work that all of you have been doing in the field of Arracacia, and I'm really happy to know the diverse scientific work that is currently being done in that field. It's a great learning experience. I have been working in this on Arracacia for a couple of years, and I got inspired from Martha to take some research questions in this field. So you already have talked a lot about Arracacia, so I thought I will just rewind a couple of steps and I will try to talk more general about the need of developing scientific tools for crops like Arracacia. So I know that I'm the only one standing between your lunch and this presentation, so I will try my level best to be very fast. And yeah, and so one of the key questions that we want to address here is food, because food matters, and we know that we all are waiting for our lunch in a couple of minutes. Now, the idea of showing you these different variety of foods that you see on this slide is because not many of us are able to consume this different variety of food on a daily basis. Interestingly, these different varieties of food that you see on the slide, they are not that expensive compared to the commercial food variety that you have in the market. Now, we think that we have plenty of food, but according to a global nutritional report that came out in 2020, there is a huge crisis of food. The report says that 1 in 9 people in the world are hungry and malnourished. And what is more surprising is that 1 in 4 children, which are under the age of 5, are stunted physically or mentally, and that affects the life cycle pattern of an individual. Sometimes you will think that perhaps they do not have access to a good diet, but interestingly, poor diets and the resulting malnutrition are not simply a matter of personal choice. The fact is, many of these people cannot afford a good quality and healthy food lifestyle. And it's very interesting that there was a study which showed that the very key question of food insecurity can provide so much of a stress in your life that it can lead to chronic diseases, which can eventually lead to more health care expenditure, and then eventually that is going to affect your household income. So access to food security can take care of a lot of stress that we all are facing these days. What is more interesting is that if you see the projection of the world population from now until 2050, we see that the least developed countries are going to have massive population explosion in the coming 10, 20 years. But if you look at the cereal crop production, you would be amazed to know that most of these least developed countries will not be able to produce enough of the cereal crop production. So perhaps they will have to depend on a lot of import. If you look at Latin America, which is shown in this very thin line, if this is the projection of the food production until 2050, it seems that even though the population of Latin America is huge, but the amount of food produced from the Latin American region is very less. And the question is there alternative that can help us to fix this problem of less food production in least developing countries, you know? Here are some interesting facts, and I'm sure some of you might be aware of this, but for those who are not aware, according to a rough estimate, according to the taxonomist, we have around 250,000 plant species, and out of that, 80,000 plant species are edible. But out of these 80,000 edible species, we only cultivate 150. And if you look at the calorie value that comes from these 150, you'll be amazed to know that only 4 plant species contribute to approximately 60 to 70% of the total calorie value of the dietary needs. And they are primarily rice, wheat, maize, and corn. Only 50 crops out of these 80,000 edible crops provide 90% of global food needs. So there is an opportunity to exploit the crops that are not exploited on a commercial level and perhaps make them mainstream so that we can address at least some fundamental concerns of the food insecurity. So one of the ways to address this food insecurity could be by improving the focus, not only on the major crops, but also on the minor crops, like, for example, Arracacia. Crops like Arracacia, they are called orphan crops, or they're also sometimes known as underutilized crops. And they're basically orphan crops are basically a diverse set of minor crops that tend to be regionally important, and they are found at different pockets of the world. But they do not receive too much of attention. I mean, I'm very glad that people are talking so much about Arracacia, but in addition to Arracacia, there are so many other unutilized crops that need our attention and that also needs a good research. So orphan crops belong to a major group of crops, which could be tuberous crop, which could be cereals, which could be fruits. So there's a variety of orphan crops that we can utilize and try to address the question of food insecurity. So this was a slide that I showed you in the very beginning, and I'm pretty sure that you know some of them. For example, nowadays, quinoa has become a very trendy food, but to be honest, 10 years ago, this was not as trendy as it has become now. It is primarily produced in the regions of Africa, and you would be amazed to know that it is very rich in riboflavins, for example. And similarly, we have other crops, and you already have talked about the nutritional value of crops like of apio. What is interesting is that most of these orphan crops, they are commercially grown in smaller countries. So still the countries which have a lot of agricultural capacity, they are not investing too much into popularizing these orphan crops. Here you can see, for example, on the extreme left-hand side, you can see some of these orphan crops, and you will see most of these countries are either in Asia or in Africa. So the agriculture technologies needs to be spent more on these orphan crops for a better production. Long time back, we had this famous green revolution, if you remember, which was mostly focused on the productivity of wheat and rice in Asia. But no such revolution has happened ever in crops like orphan crops. And I think now is a good time to have some kind of thinking and ideas which should be developed to sort of give a new paradigm to these orphan crops. Of course, we have a lot of constraints related to the properties of these orphan crops, including apio. For example, sometimes the productivity can be inferior. There could be the nutritional value of some of these orphan crops cannot be that high. We don't know if all the orphan crops have some toxic elements in it. There's always a concern of the extreme environmental conditions like stresses. But if you take all of these into consideration, there are opportunities that we can utilize for studying these neglected crops. I'm here going to give you one classical example of a commercially available crop compared to an orphan crop. So here you can see the wheat variety triticum, and here you see a crop that is grown primarily in Africa called teff. And if you look at the nutritional profile of these two crops, you will be amazed to know that, let's say, for example, the level of calcium is almost 10 times more than what you can consume from 1 gram of wheat. So they have enormous nutritional profile. What is sad, though, is that if you look at the research profile here, there has not been much focus on improving the varieties of crops like teff in Africa compared to what the research has been done on wheat. So here it's just a graph showing you the black bar is the teff, and the brown bar or the dotted bars are for the wheat. It's not that people have not started to work on these crops. There has been considerable development in the last 10, 15 years. And here is just a slide to show you that on some of these crops, some of these underutilized crops, there has been some technologies that are being developed so as to enhance the crop productivity, including genome sequencing and other omics technologies, gene knockouts. However, when you talk about the orphan crops in Latin America, one of the crops that come to our mind is the Arracacia, and there has not been too much of development in terms of genomic tools. Eduardo was talking about the development of molecular markers, but still he also at the end emphasized the importance of having more genomic data. And this is what primarily we are doing here in our lab. So I won't go in much detail about Arracacia because it has already been discussed. But one of the things that you also discussed is that the tuber of Arracacia is rich in starch, and it's very unique. Based on the literature, we have found that this tuber is being routinely used to feed infants in countries like Brazil, probably because of their very good digestive properties. So we grow Arracacia in our lab, and we try to study different tissue parts of this plant. And I'm pretty sure you're more familiar with all these different tissue parts. One of the key questions that we also want to address is the leaf, the biomass that is produced above the ground, that is primarily not used for many purposes. So we are also exploring the options to see if we can develop these biomass model for things including secondary metabolites or biofuel. So why we need to study the biology of these orphan crops? Because somehow these orphan crops, they have relation to their wild relatives. And so if we study these orphan crops, then perhaps we can understand the domestication process of these crops. These orphan crops can also provide a link to the major commercial crops, and perhaps the techniques that are available in these major commercial crops could be transferred to these orphan crops for our studies. So our project is primarily aimed at improving the genetic varieties of Arracacia and other orphan crops. And these are multiple goals that we have in our lab. We do transcriptomics and genomics. We do tissue culture and transformation. But Martha Hiraldo here is an expert on apio tissue culture, so we take a lot of input from her. We also do host pathogen interaction, and now we are trying to develop some imaging techniques using the microscopes that we have. And of course, we do all the farmer outreach activities. So today, I will only be talking about the transcriptomics and the genomic studies and the imaging techniques that we have in our lab. So we have a benchtop scanning electron microscope, and we started to investigate the leaf of Arracacia. And what you can see here is a surface of the leaf that was studied under the scanning electron microscope. And we want to identify the different types of trichomes, trichome structure that is present on the leaf. So this work is currently carried out by our PhD student, Hises, who is currently studying these structures on apio leaf. So what you see here is a 500x magnification of a trichome that is present on the upper leaf surface of an apio. And this is just a magnified version. Why we're interested in it? Because the presence of a wide variety of trichome structures could perhaps hint at the storage of secondary metabolites in these pockets. So we are really interested in identifying these trichomes and what is inside these trichomes to see if these can be potentially commercialized for applications. Another structure that we have investigated so far is the stomata. And I think most of you are aware, stomata are these tiny kidney-like structures present on the surface of the leaf that helps the plants to exchange gases. So here is a 750x magnification of these stomata, and this is 200-500x. And here you can see 25,000x magnification of one stomatal structure. So here you can see that the stomata is almost about to close. Perhaps it was collected during the time of the evening. So why we want to use a stomata as a structure? Because a stomata can serve as a molecular marker for studying stress, for example. So we can very easily, using these microscopic techniques, can study the effect of environmental constraints on the structures like stomata. Another thing that I mentioned in the very beginning was the starch. So we use this microscope to study and compare the starch of potato that you see on the left-hand side and the starch of the apio on the right-hand side. And by looking at these morphological structures, we are able to understand why this starch is easily digestible, because they have more coarse surface, and perhaps that coarse surface can provide more digestibility, digestible properties. So that's why perhaps many countries in Latin America, according to some of the published reports, they are using these starch for children and the ailing people. What it also tells us that perhaps in future, we can project the tuber as a potential alternative to potato, and which will be a healthy choice. So one of the first tools that we developed in our lab a couple of years ago was to sequence the chloroplast genome of the apio variety that is grown in the mountain region of Puerto Rico. So we potentially collected the leaves of the plant, and then we sequenced the chloroplast. So in order to sequence the chloroplast, we first isolated the DNA of the chloroplast from the leaves using a protocol that we published, and it's publicly accessible. So for those who are not very familiar with this process, I just thought I'll just put a very simplistic representation. So let us say this is the DNA of your chloroplast. then what we do is we fragment them, and then we sequence all these fragments, and then all these fragments are basically small what we call as reads. So these are basically small fragments of nucleotide bases. And then we, using bioinformatic approaches, we try to assemble them together so that eventually we can have a bigger fragment that can provide us more robust information. So using this technology of sequencing, we sequenced the chloroplast genome of Arracacia variety. This chloroplast genome has approximately 143 bases. This is publicly available, so you can read this paper, or you can also go and search for this chloroplast genome on the NCBI. It has close to 160 genes, and when we did some phylogenetic analysis, we found that this is very, very close to carrot, the Pseudocus carota, which makes sense because both of them belongs to the Apiaceae family. Now, why we wanted to sequence this chloroplast genome? Because we wanted to provide a toolkit to the researchers so that they can work on the photosynthetic genes that is present in the chloroplast genome, perhaps to alter the photosynthetic efficiency of the plant. In addition to identifying the genes, we were also able to identify a lot of molecular markers, which are also deposited in the NCBI repository. So the next project that we took was to sequence the transcriptome of the different tissues of the plant. So here, transcriptome sequencing is a method for transcriptomic studies, which basically helps us to identify the genes that are present at a special and temporal point in a different tissues. So here we sequenced 4 tissues: flower, which is very, very rare to find, because if you find the flower on an Apio plant, that perhaps signals that plant is very stressed and the plant is going to die. So we have the sequences from flower, leaves, cormel, this structure, and the storage root, because we were very interested in the biosynthesis of the starch. So this is a pipeline as to how we did the RNA sequencing. So when we analyzed this RNA sequencing result, we found that there are close to 74,000 contexts, which primarily mean that we have close to 50 to 60,000 genes in this Apio transcriptome. And we also found that out of these 74,000 contexts, approximately 65% of the contexts had a huge similarity to the carrot RNA database, which also makes sense. Only few of them has a similarity to the potato database. For 7%, we were not able to find any annotation. Perhaps these could be genes that are unique to Apio, or perhaps these could be some errors that we still need to fix. If you look at the size of the distribution of the sizes of the genes or the context, we were able to find genes that are as small as 200 base pairs to genes that are as big as 5,000 base pairs. So we were able to find a variation in the length of the context or genes. We also created a RNA browsing platform. So if some of you is interested in working or accessing the data for this RNA sequencing that we did with the Apio, it is right now it is not publicly available, but we can help you to provide the login and password. And this is something very similar to, like, if you're familiar with the NCBI database. So here you can do a lot of things, including BLAST, or including finding genes in a certain tissue that is that you want to investigate. This work was primarily done in collaboration with Céline Noy in France. She's a bioinformatician. So here I just wanted to show you like a graphical representation of the expression level of the genes. If when we compare flower and leaves, this is an MA plot. So all the red dots that you see on the upper side are the transcripts or the genes that are highly expressed. And all the red dots that you see on the lower side here are those transcripts or genes that are not highly expressed, or they're underexpressed, or they're lowly expressed. Excuse me. And what you see in the middle, the black dots, these are the collection of the genes that are not differentially expressed, meaning they do not show any strong differential expression of the genes when you compare the flower genes compared to the leaves. So we also use this RNA sequencing database to identify the genes that are possibly involved in the starch metabolism. And this is a flow chart showing you the mechanism or the pathway for the starch and sucrose metabolism in Arabidopsis. And all these different boxes, small boxes that you see here, are the genes that are involved in the pathway of sucrose and starch metabolism in Arabidopsis. We were able to identify 88 starch genes in Arracacia, and all the color boxes that you see here basically denotes to you that these are the genes that were identified in our database. When you look at the size of these 88 genes that are involved potentially in starch biosynthesis, they could be as small as 800 base pairs to as big as 3,000 base pairs or 3 kilo base pair. So Natalia is a graduate student who was working on this project of starch analysis, and she identified these 88 genes, and then she compared the expression level of these 88 genes across the different tissues that we have. So this is a heat map, and what you see in here are the names of those 88 genes, and what you see on the x-axis is the tissues. And all these single lines that you see here, they are basically genes, and the color that you are witnessing here, if a line is very dark in red, that means that the expression of the gene is very low, and if it is very bright in green in color, that means the expression is very high. What is interesting from here is that if you look at the tuber, which is primarily the site of the starch storage, we find a lot of genes that are highly expressed in the tuber tissue compared to the all other tissue, which makes sense. So then once we had this data, we were in parallel also sequencing the genome of Apio, and sequencing these genome of Apio is very important for a lot of purposes. One of the purposes that we can utilize this information is for identifying functional genes that has qualitative or quantitative traits properties. It can also help us to create a genetic resource for this plant. It can also help us to understand the evolution, how these orphan crops have evolved and how they have been domesticated from countries like Colombia to Puerto Rico, for example. And so that can also help us to sort of monitor how we can handle the best pressure if it eventually evolves, or if it eventually emerges in countries like Puerto Rico. This information of the genome can also help you to do genome targeting studies or to modify some of the traits in the plant. So we started this genome sequence project. We collected the plants from Orocovis, which is a mountain region in Puerto Rico, and we started the tissue culture. Unfortunately, in our tissue culture, we have not been able to have root structure, but we get very good shoot structure. So we use the shoot, and we extracted the DNA using the CTAB method. And we did a very large quantity of the DNA for the method that we were about to use. And we sequenced in the genome, and I will just show you very basic results that we have so far. We're still doing the analysis of those results. So we used a sequencing technology called PacBio sequencing. Eduardo was just giving you some brief about different kinds of technologies. So we use this PacBio sequencing technology. This is a technology which basically is able to sequence longer fragments of the DNA compared to the other two sequencing technologies like Illumina or BioNano, which basically sequences smaller fragments. So we were able to sequence bigger fragments, and then this is also known as long read sequencing. So we did the PacBio sequencing, and our initial results tell us that there is close to 117,000 reads. And if you look at the variation in the length of the reads, they come in different sizes, so which also tells us that perhaps the data can be trusted. So we also wanted to know what is the genome size, because even though there is some information about the ploidy level, for example, I think it's Eduardo's paper, which says that the ploidy in Arracacia is tetraploid. So we used a model to see what is the genome size, considering the ploidy level is a tetraploid. And we were able to find out that the genome size is close to 3.5 gigabases, which is huge. But it's not abnormal, considering the plant is a tetraploid. Juan Colon is a graduate student who is working on this genome sequencing project. Now, what is interesting to see here is that the size of the Arracacia genome is almost very similar to the size of the human genome. So then we do not have the information about the total number of genes, because we are still doing the processing of the assembly and the annotation. But one of the key questions that we want to know is how many protein coding genes are present in Arracacia. So based on the transcriptomic studies, we saw that we have 74,000 contigs. So which tells you that perhaps we have close to 50,000 genes. This is just an approximation. Now, if you compare the already published data from the carrot genome and we compare it with the Apio genome, we see a huge difference. The carrot is 473 megabase pairs, million base pairs, and Apio is 303.5 gigabase pairs. So it's enormous. The size of the genome is enormous. If you look at the number of the genes, it's 32,000 genes in carrot, and here in Apio, we estimate 50,000 genes. So now this gives rise to a very interesting question as to why so many genes are required and what potential functions those extra genes might be doing in Apio. Perhaps they were involved in domestication process. Perhaps they were involved in other stress-related pathways. We don't know. One thing to keep in mind when we're comparing carrot and Apio is that the published carrot genome, the tissue that they used was diploid, and one that we have used is potentially tetraploid. We are currently using a flow cytometer to estimate the ploidy level of our species. If you look at the possibility of the gene editing tools, once the carrot genome was available, a lot of genome editing tools were developed, which led to the improvement of the varieties of carrot. So in Apio, we do not have. So the availability of this genome perhaps could help us to work on genome editing tools of Apio. So in summary, we think that orphan crops can have a better future, and food security crops like Apio can be a starting point, but we also need to work on other orphan crops that are often neglected in different regions of the world. We are currently developing the genomic tools for improving the qualitative traits, and I hope that very soon we will release this data so that a community of scientists can work on these data to utilize the data for SNP or SSR markers. For example, we currently are annotating the genome, and hopefully in another 6 months, we should complete the annotation of the genome. We are also working to develop a genetic map of this species that we have in our lab. And we want to utilize all of these techniques to develop more healthy varieties, if possible, in the near future of this crop. Eventually, our goal is that once all the tools are available, we will get into the genome editing tools so that we can develop some pathogen-resistant varieties of this Apio. So I would also like to take an opportunity to tell you that I am planning to have a Latin American Orphan Crops Consortium. And this idea is coming from the fact that today we have a lot of speakers from different countries of the world in Latin America. And it is a good opportunity for us to collaborate together. Perhaps what we can do is make a list of all these orphan crops that are available in your country. And then we can enhance scientific collaborations to develop research projects which can help not only on Arracacia, but other crops that is growing in other parts of the world. So the overall goal of this Latin American Orphan Crop Consortium would be to fight malnutrition, and hopefully by utilizing different kinds of techniques, including the next generation technologies, we can develop tools that can help us to fight at least a part of the malnutrition in the coming future. So the whole idea is based on the share and collaborate ideology. So if you would like to be a part of this Latin American Orphan Crop Consortium, please don't hesitate to either contact me or contact Martha, and then perhaps we can develop this project more into a functional aspect. So this is all from me for today. I would just like to thank our Chancellor, Dr. Juan Negron, from this campus. The DNA extraction for the genome and the transcriptome RNA extraction was carried out by Rosalinda. Ramon Rivera is helping us in the bioinformatics part. Jacobo is working on the secondary metabolites from the trichomes. Natalia is working on the starch metabolism. Jesus and Juan Colons are participating in different parts of the project. I would also like to thank Jaime Sanchez from Corteva for their financial support, and Celine and Param for their bioinformatics support. And last but not the least, the USDA, United States Department of Agriculture, for all the fundings that they provide to us. And of course, thank you so much for listening to the talk, and I'm open to any question. Thank you.
Thank you, Alok.